Hi, this is Kristina Rivera, here to present from the Joint Department of Biomedical Engineering at North Carolina State University and the University of North Carolina at Chapel Hill. Today I'll be talking about tracking oxygen consumption in 3D cell cultures with phosphorescent biosensors. To do this, we were interested in recapitulating the intestinal oxygen gradient native in our small intestine, where intestinal stem cells exist at higher oxygen levels than our mature enterocytes at the tip of the villus. The anoxic lumen component is where all of our anaerobic bacteria populate. To recreate this type of physiology, we wanted to use implantable hydrogel oxygen sensors made of a tissue-like material that allow for non-invasive optical signaling. The sensors work in a way so that we can have an excitation light in a reader and detector head sitting on top of the surface, and embedded in the culture is our oxygen sensor that phosphoresces in response to local concentrations of oxygen. The phosphorescence lifetime is then recorded and correlated to local oxygen. The exploded view of our micro device is shown on the far right, and you can see that we have an acrylic setup in the center with our 5 wells, a base support, and a gas exchange channel. Above this are our rubber gasket top lid and our barb connectors for gas flow into the devices. Once the devices are sealed up using screws, we can have a gas sealed chamber and monitor the center well where the oxygen sensor is using our optics front end back to our computer outside the incubator. We found that the local level of oxygen for intestinal epithelial stem cells was much lower with increased cell densities. So for the 2 higher densities, they decreased from concentrations of 100 to 50 micromolar within 24 hours. Once fully expanded, we could adjust the level of media without having a significant influence on the local concentrations of oxygen. Lastly, we could reduce local levels of oxygen using sparging with nitrogen gas and decrease levels below 1% or 10 micromolar for continuous culture under hypoxia. Lastly, we used mature endothelial cells embedded in collagen hydrogels with our oxygen sensors to track oxygen in culture. We found that the highest density of 2 million cells per mil resulted in the smallest levels or lowest levels of oxygen below 100 micromolar. Thank you.